In this protocol, we describe a procedure containing two parts. First, a monolayer 2D keratinocyte differentiation method in vitro by contact inhibition. Second, its molecular characterization by RNA-Seq. The 2D in vitro differentiation method is straightforward and easy to perform. It can be used to study epidermal differentiation, especially when a large number of cells are necessary, for example, in epigenomic analysis. The detailed RNA-Seq analysis pipeline is clear and transparent for researchers with basic bioinformatics skills, and it can be used for any bulk RNA-Seq analysis.
When performing the differentiation protocol for the very first time, keep in mind that the seeding density can be tricky. Try out multiple seeding densities to find out which one works best with your cell line.
Begin by preparing keratinocyte growth medium, or KGM, and 500 milliliters each of proliferation medium and differentiation medium according to manuscript directions. Seed primary keratinocytes at a density of 5 to 20,000 cells per centimeter squared, and add enough proliferation medium to cover the cells. 2 days after seeding, refresh the cells with the proliferation medium. Check the cells regularly under the microscope and refresh medium every other day. When the cells reach 90% confluency, induce differentiation by changing the medium to the differentiation medium. To collect cells for further RNA analyses, wash the cells twice with DPBS and then add lysis buffer. Collect cells on differentiation.